There's an oxalotl on the pink stairs. Is an oxalotl supposed to be there? If you ask an oxalotl, nganong na ay blue nga spot? Nganong na ay blue nga spot?
DNA's ability to store, replicate, transmit, and decode information is essential for genetic function. Equally important are changes in DNA sequences, which create genetic variation necessary for phenotypic diversity, adaptation, and evolution. Gene mutations generate new alleles, driving genetic variation within populations, while mutations enable genetic analysis and help identify genes linked to specific traits. They can also cause cell death, genetic diseases, and cancer. Geneticists use phenotypic variations from mutations to track gene inheritance. Without such variability, classical genetic analysis like Mendel's experiments would have been impossible. As discussed in the previous chapters, we examine mutations in large regions of chromosomes known as the chromosomal mutations. In contrast, the mutations we will now explore are those occurring primarily in the base pair sequence of DNA within individual genes known as the gene mutations. I'm Mia Bilamoxo, and today we are going to discuss gene mutations together with my groupmates Christian Daniel Gallerport, Kian Daphne Bisande, and Hazel Grace Dolino. Next slide. So what is mutation? A mutation can be defined as an alteration in DNA sequence. Any base pair change in any part of a DNA molecule can be considered a mutation. It may comprise a single base pair substitution, a deletion or insertion of one or more base pairs, or a major alteration in the structure of a chromosome. Mutations may occur within regions of a gene that code for protein or within non-coding regions of a gene such as introns and regulatory sequences. Or mutations may or may not bring about a detectable change in phenotype. Mutations occur at about 1 in every 1 billion base pairs. Everybody has about 6 mutations in each cell in their body. So makapungutan na kasi mo self, no, like, if I have that many mutations, why don't I look weird? It is because mutations are not always seen. The affected gene may still function. Next slide. Mutations can be considered as a sustainer of life and at the same time the cause of great sufferings of many organisms. It is the source of all genetic variation, the raw material of evolution. The ability of an organism to adapt to environmental change depends critically on the process of genetic variation in natural populations, and kining mga genetic variation is produced partly by mutation. On the other hand, mutations may have detrimental effects, and it is the source of many diseases and disorders. Mutations are also useful for examining fundamental biological processes. Finding or creating mutations that affect different components of a biological system and studying their effects often lead to an understanding of the system. Next slide. For today, we are going to discuss the types of mutations, spontaneous mutations, induced mutations, and the evolutionary significance of mutations. Next slide. For the types of mutations, we have these three main types: base pair substitution, frame shift mutations, and phenotypic effects of mutations. Let's discuss first the base pair substitution. Microlesions base pair substitution is the simplest type of gene mutation. It is also known as a point mutation. It is the change of one base pair to another in a DNA molecule. So let us look at this image. Focus on yung naka-red box ha. So the original DNA sequence GGG, AGT, GTA, GAT, and CGT undergoes a change where one base or a single DNA nucleotide in a codon GTA is altered, resulting in a new sequence. This single base substitution changes only one codon, GTA, to GCA, illustrating how small alteration in a DNA sequence can lead to a change in a single amino acid during protein synthesis. Next slide. The following mutations are types of base pair substitutions. Let's first know their definitions and later we will see how they differ from each other. So first we have the missense mutation. Changes a sense codon into a different sense codon, resulting in the incorporation of a different amino acid in the protein. Second, we have the nonsense mutation. Changes a sense codon into a nonsense stop codon, causing a premature termination of that translation. Third one, we have the silent mutations. Changes a sense codon into a synonymous codon, leaving the amino acid sequence of the protein unchanged. Next slide. As you can observe from this image, we have no mutation of genes on the left side, and we are going to use this to differentiate. So for missense mutations, one nucleotide from the sense strand or coding strand of the DNA is altered from TCA to TTA, and the corresponding mRNA codons change from UCA to UUA, which the new codon encodes a different amino acid, which is leucine. So this shows a change in the amino acid sequence in the protein. As for the nonsense mutation, the DNA is changed from TCA to TAA, which the corresponding mRNA codons result in a UAA. And this new codon is a stop codon, which means there's a premature termination of translation. And this leads to the production of a shortened, potentially non-functional protein. Lastly, we have the silent mutations. A DNA change doesn't alter the amino acid sequence of the encoded protein due to the redundancy in the genetic code. For example, the coding strand of the DNA alters one nucleotide from TCA to TCG, which is the corresponding mRNA codon as UCG. This new codon encodes the same amino acid. Both UCA and UCG code for the serine amino acid, and there is no change in amino acid sequence. Next slide. For this part, we also have other terms to describe base substitution, namely the transition and transversion. When you say purines, kay mo ni siya ang adenine and guanine, and for pyrimidines, mo ni siya ang cytosine, thymine, and uracil. For transition, pyrimidine replaces a pyrimidine or purine replaces a purine, while for transversion, purine replaces a pyrimidine or a pyrimidine replaces a purine. So next slide. Now let's move on to the next type of mutation, which is the frame shift mutations. Frame shift mutations is the insertion or deletion of one or more nucleotides at any point within the gene. The frame of triplet reading during the translation is altered. It will occur when any number of the bases are added or deleted, except multiples of 3, which would reestablish in the initial frame of reading. So it is also possible that one of the many altered triplets will be UAA, UAG, or UGA, the translation termination codons. When one of these triplets is encountered during translation, polypeptide synthesis is terminated at that point. And obviously, the results of frame shift mutations can be very severe, especially if they occur early in the coding sequence. So let's look at the illustration or the image. So the loss or addition of a single nucleotide causes all of the subsequent 3-letter codons to be changed. Let's focus sa kaning naka-red box. So for the base insertion, thymine is added to the 3-letter codon, which causes the amino acid sequence to change, which is also the same for the base deletion, where thymine is removed from the 3-letter codon, altering the reading frame and may change many codons. Next slide. So we have another example, no? Let's use the sentence, the cat saw the dog, as the no mutation of DNA. So for the point mutation, also known as the base pair substitution, as what I have mentioned, is a change in a single base pair within the DNA sequence, which is analogous to changing a single letter in a word. So, ang original sentence is the cat saw the dog. So kaning substitution na, kaya nahimo siyang the bat saw the dog, which na change of C gikan sa cat to B, making it bat. For the cat saw the hog, na change ang D, which is dog, to H, making it hog. And the cat sat the dog, change of W from saw to T, making it sat. Next one is the deletion. So ang original is the cat saw the dog. Pero nakuhaan siya C sa cat. Maong nahimo siyang the ATS, AWT, HED, OG. It occurs when one or more nucleotides are deleted from a DNA sequence, and this shifts the reading frame of the codons, completely altering the amino acid sequence from the point of mutation onward. Last, we have the insertion, which is also a type of frame shift. So, no mutation is the cat saw the dog, And pag-insert sa M after sa C, name mo siyang the CMA, TSA, WTH, EDOG. And this also shifts the reading frame, leading to a completely different amino acid sequence. Next slide. Mutations in which the number of copies of a set of nucleotides increases are called expanding nucleotide repeats. This type of mutation was first observed in 1991 in a gene called FMR1, which causes Fragile X syndrome, the most common hereditary cause of intellectual disability. So looking at the picture or the table, expanding nucleotide repeats have been found in a number of other human genetic diseases, several of which are listed in the table. Most of these diseases are caused by the expansion of a set of 3 nucleotides, or known as a trinucleotide, most often C and G, where N can be any nucleotide. However, some diseases are caused by repeats of 4, 5, and even 12 nucleotides. In several diseases, example is the Huntington disease, the nucleotide expansion occurs within the coding part of a gene, producing a toxin that has extra glutamine residues, which is the amino acid encoded by CAG. Next slide. So we already reached for the third main type of mutation, which is the phenotypic effects of mutation. Non-mutations. Mutations that result in a complete loss of function, meaning the protein encoded by the gene either isn't produced at all, or if produced is completely non-functional. It completely eliminates a gene's function, leading to no functional gene product being produced. Next, we have the recessive mutations. Result in a wild-type phenotype when present in a diploid organism and the other allele is wild-type. So for the mutant phenotype to be expressed, both copies of the genes in a diploid organism must have the mutation. Next, we have the dominant mutation. Results in a mutant phenotype in a diploid organism even when the wild type is also present. So a single copy of the mutated gene is sufficient enough to cause a noticeable effect or trait. Often the disease, as a mutated allele dominates the normal allele, and it has 2 types, the haploinsufficiency and a dominant gain of function. When you say haploinsufficiency, it occurs when a single wild-type copy of the gene does not produce enough gene product to bring about a wild-type phenotype. So, ang example ani sa humans kay Marfan syndrome, because in this case, as a result of the loss of function mutation in one copy of the FBN1 gene. So next, we have the dominant gain of function. This results in a gene product with enhanced, negative, or new functions. This may be due to a change in the amino acid sequence of the protein that confers a new activity, or it may result from a mutation in a regulatory region of the gene, leading to the expression of the gene at higher levels or at abnormal times or places. Next slide. Another phenotypic effect is the visible mutations. Monishaang mutations are recognized by their ability to alter a normal or wild-type visible phenotype. So, example, all of Mendel's P characteristics and many genetic variations encountered in Drosophila fit this mutation, since they cause obvious changes to the morphology of the organism. Next, we have nutritional or biochemical mutations. In bacteria and fungi, typical nutritional or biochemical mutations result in a loss of ability to synthesize an amino acid or vitamin. In humans, sickle cell anemia and hemophilia are examples of diseases resulting from biochemical mutations. Although such mutations do not always affect morphological characters, they affect the function of proteins that can impinge on the well-being and survival of the affected individual. Next, phenotypic effect is we have the behavioral mutations. So, a genetic mutation that alters genes controlling an organism's behavior, leading to changes in their behavioral patterns. So, mutations can alter the way genes function, leading to changes in the way an organism processes information, interacts with its environment, and responds to stimuli. So, for example, the mating behavior of a fruit fly may be impaired if it cannot beat its wings. So, however, the defect may be in the flight muscles, the nerves leading to them, or the brain where the nerves impulses that initiate wing movements originate. Next is we have the regulatory mutations. affect the regulation of gene expression. It is a mutation in a regulatory gene or a gene control region can disrupt normal regulatory processes and inappropriately activate or inactivate expression of a gene. So, for example, a regulatory gene controls the lac operon. Ang lac operon is a set of genes for uptake and metabolism of lactose. So, mutations within this gene can produce a regulatory protein with abnormal effects on the lac operon, affecting transcription initiation, mRNA splicing, and mRNA stability, and may occur in splice junctions or promoters. Next slide. So the lethal mutations adversely affect a gene product that is essential to the survival of the organism. So a lethal mutation is a change in the DNA sequence, a gene mutation that, when expressed, leads to the death of an organism or a substantial reduction in its ability to survive and reproduce. So, for example, Tay-Sachs disease and Huntington's disease are caused by mutations that result in lethality, but at different points in the life cycle of humans. Next, we have the conditional mutations. Conditional mutations affects on the organisms in ways that depend on the environment in which the organisms find itself. So they are present in the genome of an organism, but can be detected only under certain conditions. So one of the best example for conditional mutation is the temperature-sensitive mutations. It is a genetic changes that cause a mutant phenotype at high or low temperatures while retaining a wild-type phenotype at normal temperatures. Next, we have the neutral mutations. So it occurs either in a protein coding region or any part of the genome. So example is a neutral mutation within a gene may change a lysine codon, AAA, to arginine codon, AGA. So the two amino acids are chemically similar. Therefore, this change may be insignificant to the function of the protein. Because of the eukaryotic genomes consist mainly of the non-coding regions, the vast majority of mutations are likely to occur in the large portions of the genome that do not contain genes, and these may be considered neutral mutations if they do not affect the gene products or gene expression. Next slide. Now we have the forward and reverse mutations. A mutation that changes the wild-type phenotype to a mutant phenotype is what we call a forward mutation, whereas a reverse mutation or a reversion changes the mutant phenotype back to the wild-type phenotype. Next, we have the suppressor mutations. It is a genetic change that hides or suppresses the effect of another mutation. So, a suppressor mutation occurs at a site that is distinct from the original mutation. Thus, an individual with a suppressor mutation is a double mutant, having the original mutation and the suppressor mutation but exhibiting the phenotype of a non-mutated wild type. So, na siya duha ka types, the intragenic and the enterogenic. When you say intragenic, it suppresses the effect of an earlier mutation within the same gene. But when you say enterogenic, it suppresses the effect of an earlier mutation in another gene. Next slide. So, we have another image. So, let us look at this image. So, a forward mutation sa kaning number 1 changes the wild type phenotype into a mutant phenotype, which is a fruit fly with red eyes to white eyes. For reverse mutation, as you can see in number 2, it is exactly the reverse of the forward mutation, where it restores the wild type gene and phenotype, which is from white eyes to red eyes. And next, we have the suppressor mutation, and it occurs at a site different from that of the original mutation. So, mutation A negative causes white eyes, and the second mutation, the mutation B negative at the different gene site, suppresses the effect of mutation A negative. And with this, it generates an individual with both the original and the suppressor mutations, but with a wild type phenotype. So that's all for my part. Let me call on Mr. Gallerport to continue the discussion on the categories of mutation. Thank you so much.
To continue, the mutations are also classified based on their locations. These include somatic mutation and germline mutation. When we talk about somatic mutation, this refers to any cells in the body except germ cells, like for example, imong skin cells. Ang mutation na ni-arise imong somatic cells are not transmitted to future generations. On the other hand, germline mutation arise in the cells that ultimately produce gametes. A germline mutation can be passed to future generations, producing individuals that carry the mutation in all their somatic and germline cells. This also correlates sa concept sa katong X and Y linked na mutations. Next slide please. So here we can see that the diagram represents the somatic and germline mutation. The somatic mutation in the upper side of the diagram undergo mitosis and mutant cells arise. On the other hand, through sexual reproduction and meiosis, the germline mutation in the lower side of the diagram pass approximately half the members of the next generation. These result in a population where approximately one exhibits a mutation and the other does not carry and exhibit no mutation. As we learn about mutation, let us now understand how rates of mutation differ from one another. So what is mutation rate? Mutation rate is the likelihood that a gene will undergo mutation in a single generation or in forming a single gamete. It is generally expressed as the number of mutation per biological unit, which may be mutation per cell division, per gamete, or per round of replication. So the differences in mutation rates among species may be due to differing abilities to repair mutations, unequal exposure to mutagens, or biological differences in rates of spontaneously arising mutations. Even within a single species, spontaneous rates of mutation vary among genes. The reason for this variation is not entirely understood, but some regions of DNA are known to be hotspots of mutations. When we say hotspot for mutation, it refers to the degree of susceptibility of some DNA sequence to mutate. For instance, the mutation rate of Duchenne muscular dystrophy in humans are about 9.2 times 10 to the power of negative 5, which is much lower compared to hemophilia A with about 3.2 times 10 to the power of negative 5 per gamete, according to the table presented. Now we are done discussing the types of gene mutations. Let us now dig deeper if such mutation occurs spontaneously or is it because of other factors that induce such mutations. First, let us differentiate spontaneous mutation from induced mutations. Next slide please. Spontaneous mutations are changes in the nucleotide sequence of genes that appear to occur naturally. No specific agents are associated with their occurrence and they are generally assumed to be accidental. In contrast, induced mutation results from the influence of extraneous factors and may be the result of either natural or artificial agents. In this diagram, for example, induced mutation happen from exposure to radiation, chemicals, or UV light, in comparison to spontaneous mutations where it happened due to errors in DNA repair, replication, or transcription. Now let's focus on spontaneous mutations with its 6 categories. So first is DNA replication errors and slippage. DNA replication errors happen when first mispairing and point mutation and second is introduction of small insertion and deletions. First is mispairing and point mutations. So it happens when DNA polymerases occasionally insert incorrect nucleotides during replication of strands of DNA. If these errors are not detected and corrected by DNA repair mechanisms, they may lead to mutations. The second one is introduction of small insertion and deletions. This occurs when one strand of the DNA template loops out and becomes displaced during replication, or when DNA polymerase slips. Insertion and deletion may lead to frameshift mutations or amino acid insertion or deletion in the gene product, as discussed previously. Later, there will be a diagram representing this one. So now let's proceed to replication slippage. When we talk about replication slippage, it occurs anywhere in the DNA but distinctly more common in regions containing tandemly repeated sequences. Tandemly meaning they are a group of two or more nucleotides arranged one behind the other acting in conjunction. Next slide please. Here it shows a possible strand slippage, insertion and deletion. In the left side, for example, the new synthesized strand with the adenine nucleotide loops out in 3' from the sequence, resulting in the addition of one nucleotide. In contrast to the right side of the diagram, the looping out of the thymine nucleotide in the template strand causes deletion or emission to the new strand. Second is tautomeric shifts. Tautomeric shifts are shifts and changes, especially in the biological important tautomers in its bonding structure, allowing hydrogen bonding with non-complementary bases. There are what we call tautomeric forms. Basically, next slide please, tautomeric shifts are altering the base pair, keto-enoforms of thymine and guanine, and the amino-iminoforms of cytosine and adenine. As we all know, the standard base pairing is thymine and adenine, as well as cytosine and guanine. However, in the anomalous base pair arrangement, It will be thymine with guanine and cytosine with adenine. Next slide, please. So in here, it illustrates how tautomeric shift happen. Instead of A pairing with T, through tautomeric shift, anomalous CA base pair are formed. As these undergo semi-conservative replication, the tautomeric shifts back to its amino form, leading to one replicate with no mutation, while the other one has transition mutation, which is the CG mutation. Why is C and G considered a transition mutation? This is because in the first semi-conservative replication, it should be T and A base pair. This change led to the second semi-conservative replication to replicate C that pairs with G when in that section of the sequence it should be T and A base pair. Another question in mind would be, why does tautomers shift back? This is because they are interchangeable forms due to chemical bonds and they rearrange many times. Now let's proceed to the third one, depurination and deamination. Depurination is the loss of one nitrogenous bases in an intact double helical DNA molecule. These bases may be lost if the glycosidic bond linking the 1'C of the deoxyribose and the number 9 position of the purine ring is broken, leaving an apurinic site on one strand of DNA. If the apurinic site are not repaired, DNA polymerase may introduce a nucleotide at a random at that site. Next slide, please. Due to depurination, as you can see, apurinic sites are created. This site cannot provide a template that complements its base pair during replication, leading to an incorrect base, usually adenine, to complete a sequence. In the next round of strand replication, both strands with apurinic site and the mutant one serves as a template for a series of gene sequences that leads to the permanent mutation of the genes. Now let's proceed to deamination. Deamination happens when amino group in a cytosine or adenine is converted to a keto group. In these cases, cytosine is converted to uracil and adenine is changed to hypocytine. Next slide, please. As you can see, the major effect of these changes is an alteration in the base pairing of these two bases during DNA replication. When adenine is deaminated, the original AT pair is converted to a GC pair because hypocytine pairs naturally with cytosine. Deamination may occur spontaneously or as a result of a treatment with chemical mutagens such as nitrous acid. Here's another diagram showing deamination. Now let's proceed to the fourth one, oxidative damage. DNA may also suffer damage from the byproducts of normal cellular processes. These byproducts include reactive oxygen species, which are electrophilic oxidants that are generated during normal aerobic respiration. Reactive oxidants are also generated by exposure to high-energy radiation, which produces more than 100 different types of chemical modification in DNA, including modification to bases, loss of bases, and single-stranded breaks. Now let's proceed to the fifth one, mispairing due to other structures. Mispairing often arises through a wobble in which normal protonated and other forms of the bases are able to pair because of the flexibility in the DNA helical structure. These structures have been detected in DNA molecules and are now thought to be responsible for many of the mispairing in replication. In relation to wobbling, the last one is incorporated and replicated errors. Incorporated errors happen when a mispaired base has been incorporated into a newly synthesized nucleotide chain. As you can see in step 2, thymine pairs with guanine due to wobbling. Replicated errors, in simpler terms, are errors that create a permanent mutation. In step 3, all the base pairings are correct. However, it utilizes the wobble strand in step 2. As this replicates, there is no mechanism for repair system to detect the error and transition mutation happens.
So, mutations are changes in the DNA sequence, and this can occur naturally or what we call as spontaneous mutations, or can be triggered by external factors known as induced mutations. So speaking of induced mutation is a result from the influence of extraneous factors and may be the result of either natural or artificial agents. So geneticists also harnessed some mutagens to analyze genes and gene functions. To make it short, so induced mutations happen when external agents called mutagens alter the DNA. So, next slide please. Mutagen, as we know, it's a plethora of agents and has the potential to damage the DNA and can cause mutations. There are some examples of mutagens. We have fungal toxins, cosmic rays, ultraviolet, or what we call as natural component, or we also have industrial pollutants, medical rays, or chemical in tobacco smoke, or what we call as a natural or human-made. So, we also have types of mutagens na atay physical o chemical mutagens. So, for physical mutagens, we have ionizing and non-ionizing agents. And for chemical mutagens, we have agents that alter base pairing and DNA structure. Now, let's talk about the physical mutagens, or what we call as the ionizing radiation. So what is ionizing radiation? Ionizing radiation is a type of energy that can break the chemical bonds and can cause mutations. It includes high energy frequencies such as X-rays, gamma rays, and cosmic rays. And radiation sa kaninga mga energy kay mag-vary siya inversely with their wavelengths, corresponding to their higher energy levels. And this form of radiation is capable sad siya nga makapenetrate deeply sa atuang tissue and can cause ionizing of molecules nga ilahang ma-encounter. That is the ionizing radiation. And also, as it penetrates the cell, ang katong mga stable nga mga molecules and atoms are transformed into free radicals. So, free radicals kay kanang mga highly reactive chemical na sila with unpaired electrons. And also, these free radicals can cause several types of DNA damage, including altering purines and pyrimidines sa atong DNA, or makabreak siya sa phosphodiester bond, which can lead to strand breaks. So, it can also kanang chromosomal aberrations, including deletions, translocations, and chromosomal fragmentation. There are researches nga nakademonstrate sa di sa linear relationship between sa ionizing radiation ug sa mutation rate. Nya kaning each of the radiation exposure nila kay each doubling nga radiation exposure nila kay twice as many mutations sad ang mahitabo. Now there are other factors pod. We also have, diba, anak ko ganiha, we have UV radiation and pyrimidine dimers. So in terms of the UV radiation and pyrimidine dimers, so UV light is less energetic than ionizing radiation, but still mutagenic siya. Dili siya maka-remove of electron pero maka-form siya abnormal chemical bonds between sa yang adjacent nga pyrimidine bases or la thymine or cytosine. So in terms of pyrimidine dimers, ang thymine dimers are the most common. Nya ang kaning mga dimers mo distort sa DNA, mo block sila sa replication ug sa transcription. And if ang replication kay ma-block, of course, ang cell kay mamatay, which is why UV light is used as sterilizing agent. So as we know, UV radiation is a physical mutagen that primarily cause pyrimidine dimers, especially thymine dimers. And these dimers are distort DNA structure, blocking replications. So as we see sa figure 14.8, ang induction niya of thymine dimer by UV radiation nakalead siya sa distortion of DNA, and ayang covalent crosslinks occur between the atoms of the pyrimidine ring. So makita nato sa picture, dimer formed between the adjacent thymidine residues along DNA strand. We also have the SOS system in bacteria. So some bacteria have SOS repair to bypass sa replication blocks tungod sa caused by DNA damage. However, ang kaninga system is error prone because wala siya nag-follow og strict nga base pairing rules. So more siya trade-off, the cell survives but at the cost of high mutation rates. And now, so humanata sa physical mutagens, let's go to the chemical mutagens. Now, chemical mutagens. So chemical mutagens are substances nga ni-interact sa DNA to induce mutations. Kani nga mga agents are classified sila into several categories. We have base analogs, alkylating agents, intercalating agents, and adduct forming agents. Now, ato na sila itag-sa-tag sa discuss. So what is base analog? So base analog, similar structure sila sa normal DNA bases and can be incorporated into DNA during replication. However, mag-exhibit sila altered base pairing properties leading to mutations. Ang example ani kay kaning 5-bromouracil that behaves as thymine analog and in its common keto-form. Ang iyang base kay nakig-pair siya with adenine, but in the rare enol form, it pairs with a guanine leading to AT to GC transitions. And next slide, we're gonna talk about the alkylating agents. Now, ang alkylating agents kay ma-modify ni siya sa DNA by adding alkyl group, let's say methyl ni siya or ethyl sa nucleotide bases, thereby maka-adhere siya sa base pairing properties. And atong examples ani we have ethyl methylene sulfonate, or what we call as EMS. We also have deamination, and we also have hydroxylamine. So I'm gonna talk about the meaning of each pair, and then sa next slide makita nato kung unsa akong pasabot. So the first one is the ethyl methane sulfonate, or what we call as EMS. So ang kani, it alkylates guanine at position 6 nga yang i-convert to 6-ethyl guanine. So which then pairs with thymine instead of cytosine. So ang transition mutation niya kay nahimo na nun siyang GC to AT transition. And then the next one is deamination. So actually ang deamination can happen ra siya naturally or can be caused by nitrous acid. So nitrous acid kay i-change niya ang cytosine to uracil or the CG to TA nga pairing, adenine to hypoxanthine like TA niya ma-pair na sa TA to CG, and guanine to xanthine, which can lead to ang pairing kay makuha na siya, mahimo na siyang CGTA or like a change, since it only causes transition nya. Ang nitrous acid can also reverse the mutations pod. And last one is ang hydroxylamine. So hydroxylamine is a specific mutagen nga nag-add og kanang hydroxyl group sa cytosine nga ma-convert siya into hydroxylaminoscytosine, and kini kay maka-increase siya sa chance of mispairing sa adenine instead of guanine. So ang mahitabo, ang CG will be paired to TA. So transition, CJ to TA transition rather. Since it only affects cytosine man, it cannot reverse the mutation it creates, unlike sa deamination nga ang katong nitrous acid can reverse these mutations. Now, sa next slide, makita nato nga ang first picture, which is letter A. That is what we call as the ethyl methane sulfonate. Mao na iyang pairing. The second one is the deamination, the letter B, and the letter C is the hydroxylamine. Now, let's talk about intercalating agents. So, intercalating agents like proflavin, acridine orange, or ethidium bromide, or dioxin, ilang insert ilang self between sa DNA bases nga makadistort sa structure. Unya kani nga disruption kay maka-lead siya to single nucleotide insertions or deletions during replications. Maopod ning often nga maka-cause og severe frameshift mutations, since maka-create man siya both additions and deletions, and kini nga mga agents can also reverse their own mutations. Makita na nato siya sa figure 13.9, or like let's say next slide. Makita nato siya next slide. 13.19, the first one, this letter A, the produced mutation by sandwiching themselves or intercalating between adjacent bases in DNA, nakadistort siya the three-dimensional structure of the helix and causing single nucleotide insertions and deletions in replication. The second figure shows that these insertions and deletions frequently produced frame shift mutations, so the mutagenic effects of intercalating agents are often severe. So additions and deletions can reverse mutations as they produce. So this picture shows that ang intercalating like ethidium bromide insert themselves between DNA bases and nakadistort siya sa DNA structure. Nga nakakause siya sa mistake during replication, and then nakalead siya insertion or deletions or the frame shift mutations, and this mutation can naka-change siya sa the way the genetic code is read. So often making proteins non-functional. So if not repaired, they can lead to disease like cancer.
So I'm just gonna give you a quick recall, no? Kaya the difference between induced and spontaneous mutations is, aside from their mechanism, in which spontaneous is naturally occurring over time, while induced gani, meaning in this context, gibutang siya to trigger, so there can be an external intervention. Both ipasabot, induced mutation is an intentionally triggered mutation. Let's proceed to the third type of induced mutations, which is by transposable elements. When we think about genes in a normal mechanism, diba allele arrangements can be calculated. Unsa may middle gene, unsa may relative distance between the genes. And we would normally comprehend nga the DNA sequence already has a structured arrangement, thus reflected by our phenotypes, diba? Karon, there can be instances when these stable arrangements can be disrupted. Sometimes, These disruptions are caused by transposable elements or the jumping genes. Muambak gani, the landing represents the insertion or ang pag-insert ana nga gene into the sequence. Mao na ang makakause og mutation. Now, di ba ni-mention ko earlier about induced and spontaneous mutation? Transposable elements can actually cause both because these elements are already naturally in the genome. So, pwede siya, number 1, triggered by external factors, and pwede pod number 2, out of natural transposition or the movement from one location to another. Barbara McClinton in the 1940s discovered transposable elements through her corn research. Normally, diba, corns are solid colored, usually yellow. When she observed the spotted color variation, she hypothesized that there may be genetic rearrangements, which led her to discovering transposable elements. Transposable elements have two types. Class 1 is the retrotransposons. Take note, transposable elements are also called transposons. Retro, in general, means imitative or musundog sa style of the past. That is why very justifiable ang iyang copy and paste mechanism. So let me run this through. Retrotransposons move via RNA intermediate, require reverse transcriptase enzyme from RNA to DNA, and stays in place while a new copy inserts elsewhere. Diba, same ra kung mag-control C ta sa atong documents. Mabilin si original and sa laing lugar ta mag-paste. For DNA transposons, or ang class 2, they move via DNA intermediate, require transposase enzyme, and the original element moves instead of copying itself. That justifies cut and paste mechanism because ang original transposon man ang galihok. For further representation of the two mechanisms, especially for visual learners like me, let's try to understand the breakdown. For retrotransposons, as you can see there, the original sequence, it will have a staggered cut, which creates a gap where the retrotransposon can insert. Unsaon man pag-copy, this original transposon isn't directly copied as a DNA molecule. It's initially copied as an RNA molecule, just like how a gene makes messenger RNA for protein synthesis. An enzyme called reverse transcriptase takes the RNA molecule and converts it into DNA. Needs siya ma-convert into DNA because an RNA molecule cannot directly insert into a DNA sequence, diba? Tungod sa ilang structural and chemical differences. So going back, original transposon is copied in the form of an RNA molecule and then transcribed into DNA by reverse transcriptase. The new DNA copy or the new transposon inserts on another site, and DNA repair happens. As you can see, the sequence on your left of the transposon CCGTAA is duplicated as seen on your right. That is part of the DNA repair mechanism, and we'll discuss that later on. Next, we have the cut and paste mechanism. DNA transposon from the name itself is already in the DNA sequence. This by then is cut from the original sequence, as you can see in number 2, and inserts into a new site. Unlike retrotransposon that's transcribed into RNA back to DNA, this is constantly in DNA form already, since cutting means having and moving it as it is, rather than synthesizing a new copy. And DNA repair, of course, naturally happens. Retrotransposons use replicative transposition only, while DNA transposons can be replicative or non-replicative, meaning pwede siya both. Let's have here a simplified comparison between replicative and non-replicative. Process-wise, replicative inserts a new copy while the original stays, whereas non-replicative excises or cuts and moves to a new site. There is an increase in copy numbers sa replicative, of course, because duplicated baya ang original transposon, while sa non-replicative, kaya there are no changes. Replicative needs DNA replication, while for non-replicative, only flanking direct repeats are replicated. Unsa ning flanking direct repeats? Flanking direct repeats are short, identical DNA sequences that appear on both sides of a transposable element after it inserts into a new location in the genome. Sa duha ka mechanism ganiha, unsa ganito mahitabo after insertion? DNA repair. The flanking direct repeats are part of DNA repair. Let's take a look at here. As mentioned earlier, transposition starts with staggered cuts. A staggered cut is when transposase cuts the DNA strands at different positions, not straight across. Imagine using scissors to cut a ribbon in a zigzag pattern instead of a straight line. Mao na siya. Eventually, transposon insertion happens. The staggered cuts leave short, single-stranded pieces of DNA. DNA repair will aim to fill those gaps by generating flanking direct repeats. Flanking direct repeats are important for two main reasons. Number one, it ensures that the DNA sequence remains intact after transposition, and of course, marks the location where a transposable element has been inserted into the genome. We also have terminal inverted repeat. This is not part of the DNA repair. This is specific for the movement of the transposon. They are inverted complements of one another. Let's take for example here, as you can see on the screen, on the 5' end, or kanang nakita ninyo nga 5 apostrophe. That's read as 5'. ACAG becomes the inverted GACTTGACA. Still on the 5' end. Unsa may meaning aning 5' ug 3'? Diba, DNA has two strands that run in opposite directions. As you can see on the left of the PowerPoint, with a 5' phosphate end and a 3' hydroxyl end. So, ang duha ka strand, ibutang nato, strand 1 is 3 to 5' and strand 2 is 5 to 3'. So, bali-bali sila. Transposase can only and primarily recognize specific 5' to 3' sequences. Usara ka strand iyang ma-recognize. This is why terminal inverted repeats exist. They ensure that the enzyme properly identifies and binds to both ends of the transposable element. In simpler terms, mao ni rason nganong ang duha ka strand sa DNA sequence ang ma-insertan sa transposon. If ibutang nato, wala niya ang terminal inverted repeat, ang usara ka strand ang ma-recognize sa transposase. If kana ang mahitabo, transposition will fail and can cause an unstable single-stranded DNA. That is why understanding these mechanisms, including the components, is very important if you are learning about induced mutations caused by transposable elements. That's it for the discussion on transposable elements. Let's proceed to the evolutionary significance of mutations. Spontaneous mutations are important in evolution because they create genetic diversity which natural selection acts upon, survival advantages under environmental pressures, and neutral mutations accumulate over time, potentially leading to evolutionary shifts. By introducing new traits into a population, mutations provide the raw material for natural selection to act upon. Diba? Survival of the fittest. These new traits from mutation can make an organism fit to survive. Beneficial mutations can enhance survival and reproductive success, leading to adaptation to changing environments. It's a domino effect. Without spontaneous mutations, populations would lack genetic variability, making them less adaptable to environmental pressures and more vulnerable to extinction. Example, genetic drift in small populations. Spontaneous mutations become more common in isolated populations due to random chance, leading to evolutionary divergence. Diba, as said, unique beneficial traits may be obtained through this evolution. For induced mutations, they can drive rapid adaptation by introducing genetic changes. This is because induced mutations can selectively introduce genetic traits that can immediately enhance an organism's ability to survive in changing environments. Also, dili rin siya for individual nga genetic changes because they may cause large-scale genetic changes, influencing evolutionary pathways. Large-scale genetic changes can be caused by disrupting multiple genes or regulatory sequences through DNA breaks, chromosomal rearrangements, or even, as mentioned earlier, transposon activation. These alterations can significantly impact gene expression and function, influencing evolutionary pathways by introducing new traits or adaptations. And they are often linked to human-driven environmental changes such as pollution and antibiotics. An example can be radiation-induced mutations. Radiation exposure has led to genetic changes in plant populations, affecting their growth and adaptation. Indeed, change is inevitable. And when we talk about mutations, there will always be pros and cons. Dealing with it, we already have our best weapon, which is science. And it's up to us on how we digest that knowledge and spread it to other people. Thank you for listening.